Hello, everyone. My name is Dr. Marwan. I'm a lecturer in the Faculty of Pharmaceutical Sciences, UCSI University, Malaysia. My research area in nanomedicine, and I'm interested in developing a novel strategies for targeted delivery of cancer therapeutics. In today's presentation, I'm going to share with you my recent work in this area by using functionalized nanoparticles for targeted delivery of cancer therapeutics. First, I'm going to discuss how this targeting complex was designed virtually by using in silico techniques. The interaction of the molecules used to develop this complex was studied first virtually, and also the targets, the possible targets for these molecules and for the overall complex was also predicted by using the in silico techniques. Then next, the complex was developed in the lab and then evaluated in vitro for different properties, for example, its stability and the drug release from the complex, and also the cytotoxicity of the complex in comparison to bio drug. So the objective here is to improve the selectivity of the anti-cancer drug towards the cancer cells in order to improve the activity of the anti-cancer drug and also to reduce the side effects. Then the targeting technique also was tested in vivo to ensure that the complex can be delivered to the targeted site. Then finally, the complex was developed as a capsule, and that capsule also was coated with a specific coat, which is the enteric coating, and also to deliver the content of the capsule to the targeted area. And in this case is colon area. So we targeted the colon cancer. The drug used in this study is everolimus. Everolimus is mTOR receptor inhibitor. It's known to have anti-cancer activity and could be effective against colorectal cancer, but the mechanism behind this activity is still unknown. And it's known also to produce severe side effects. For example, stomatitis, mouth and lip ulceration is always observed. Around 44% of the patients develop these side effects. So again here, by improving the selectivity of the everolimus towards cancer cells, we could be able to reduce the side effects of that drug. So the study started with, as I mentioned, with the in silico studies to predict the targets of the individual molecules and the overall complex, and eventually to design the anti-cancer complex virtually and then test it in vitro, in vivo, and develop as a product, in this case as a capsule. So in the development stage, this drug is known to have very poor aqueous solubility. So it's not easy to work with this drug. So in this case, we choose the beta-cyclodextrin as an inclusive compound to incorporate the drug into the inner part of the cavity of the beta-cyclodextrin, because the cavity of beta-cyclodextrin is known to be hydrophobic. So the drug could be incorporated into the cavity of the beta-cyclodextrin, and then the surface of the exterior of the beta-cyclodextrin is known to be hydrophilic. So in this way, we improve the aqueous solubility of the everolimus, and it become easier to work with. Because in the next step, we need to incorporate the targeting moiety and then to formulate as a capsule. So in the step of incorporating the complex, let's call it here the inclusive complex, because it's contain the drug within the beta-cyclodextrin. So in the step of incorporating the targeting moiety with the complex, it was very simple because we improved the solubility of the anti-cancer drug. So Here, we just simply dissolve the inclusive complex into aqueous solvent, and then with the targeting moiety, then with electrostatic interaction, the targeting moiety was able to coat that complex. We use here FGF molecule as a targeting moiety, which is fibroblast growth factor. So why we choose this? Because we wanted to use it as a targeting towards the receptors that is already known to overexpressed on the colon cancer cells. So these receptors are overexpressed. So this molecule could target the anti-cancer drug toward these receptors and make that anti-cancer agent more selective to cancer cells. Why? Because the cancer cells overexpress the receptors, FGF receptors. So first we tested, again, this is still we are in the virtual part of this study, so the in silico part. So we tested the interaction between the cyclodextrin with the FGF and with the drug. So to ensure that the complexes could be formed in the lab or developed in the lab. So here also we found that cyclodextrin is able to bind with the FGF. Also, everolimus also can bind to FGF. This is very important. Later we know why it's important. But this is also confirmed that the complex is possible to form virtually and also in the lab. And we compare here with heparin, because heparin is known to interact with FGF in order to activate FGF and then produce its activity by activating the cell signal. So here the interaction is compared to heparin to see how strong the interaction between these molecules, and then we can predict the possible activity of the complex after all these compounds bind together. So we can see here, this is a possible of activity of the complex. So here we can see at the left side, heparin, this is the normal process. Heparin would interact with several molecules of FGF, then FGF would interact with the receptor to produce its action, to activate cell signals. But in our case, that FGF will be interacted with everolimus and or beta cyclodextrin. So here we predicted that this will inhibit the interaction of FGF with heparin. That means FGF will not be active, but still it could bind to its receptors. But it will bind to the receptor, but it will not produce any effect. So that means it will not activate the cell signals. So in this way, we will be able to utilize FGF as a targeting moiety, but it will not produce any cell signal. So because FGF is known to produce or to enhance or to improve the cell growth, right, or cell repair. So in this case, it could also improve the growth of the cancer cell. So we want to neutralize that. So this is how we neutralize the effect of the FGF. So here again, FGF will not have any or they will have very minimal effect on the growth of the cancer cell, but it will produce a targeting effect. So then after forming the complex virtually, then the complex was developed in the lab, then tested using colon cancer cells. In this case, we use COCO2 cell line. So again, the complex was formed from everolimus anti-cancer drug, beta cyclodextrin, which is used to encapsulate the everolimus to enhance its aqueous solubility. So this is the main reason we use beta cyclodextrin here, but the whole complex is also coated with the FGF as a targeting moiety. So this is the complex, and then the complex was tested into Kakutu cell line and compared with the pure drug, and it was observed that it significantly enhanced the activity of the anti-cancer drug. So again here, the theory behind this is that enhancing the selectivity means the drug will be accumulated at the target site and produce the maximum effects needed to inhibit the growth of anti-cancer cells and minimize its effect on the normal cells. That means reducing the adverse effect that we mentioned earlier. So we can see here the maximum amount used from the complex, the concentration, I mean, of the anti-cancer drug within the complex, in this case, 13.04 micromolar, produced the maximum inhibitory effect on the Kakutu cell line when compared, for example, to pure drug. Okay, so the IC50 dropped from 9 micromolar to around 3 micromolar. So it's a significant improvement in the anti-cancer activity for the complex. And also, one of the studies, the visual studies conducted here to predict the targets of not just the complex and also the individual molecules used to formulate the complex. Why we did that? Because we need to know the potential mechanism of action of this complex. So how this complex could produce its activity. Is it just because the anti-cancer drug will bind to its receptor, mTOR receptor, and produce its inhibitory effect and reduce the growth or lead to cell death eventually, or it's a combination of multiple effects due to the molecules used to formulate this complex? And actually we predicted that it's the second scenario. Why is that? Because beta-cyclodextrin, according to this study, target prediction study, we found that it could target galactin-3 protein. So this protein is also have a known effect to facilitate the cell adhesion, cell signaling, and also migration of the cancer cells. So if we can also inhibit this protein, we can also inhibit the cell adhesion, migration, and so on. So it could contribute in the activity of the overall complex. The second prediction here also is that the beta-cyclodextrin, as we mentioned, it can bind to FGF. So FGF, it's growth factor. So again, also FGF could have effect by facilitating the growth of the cancer cells. So here, if the beta-cyclodextrin can inhibit FGF, also can reduce the possibility of the growth of the cancer cells. So this is how we the assumption of our study is that the effect of the complex, not just because of the anti-cancer drug, it's a combination of all these effects produced or resulted in the improvement of the anti-cancer activity of the complex. As we can see here, for the left at the left side here, this is the result for the cytotoxic activity on the Kakutu cell line. As we can see here, the maximum cytotoxicity was observed with the complex, as we can see here, FGF with beta-cyclodextrin and everolimus, so produce the maximum cytotoxic activity when compared to, for example, everolimus, the pure drug. Okay, and compare also to, this is also very interesting here. So this one is, it was inclusive complex, but without the FGF. Also, the cytotoxicity is less compared to this complex. And this is here the targeting. Because of the targeting, FGF is actively target the complex or the anti-cancer drug to the receptors or the cancer cells. So that is resulted in increase in its activity. The second study here is the migration. So the migration here, as I mentioned earlier, so we assume here that by inhibiting galectin-3, we could be able also to inhibit the migration of the cancer cells. And as we can see here also from this graph, that the complex here is also significantly inhibit the migration of the cancer cells when we compare to, for example, the control, negative control, and also the positive control with the drug. So this is also here another example for how this complex could improve the activity of the drug. So in this case here, by also inhibiting the migration of these cells. So by inhibiting the migration of these cells, we could also improve the activity of the anti-cancer drug. Another study also was to study the effect of the complex on the mobility of the cells. So here also mobility could be translated on the migration or in general, the invasiveness of these cells, the cancer cells. So as we can see here, we use the scratch test to study the effect of the complex on the mobility of the cancer cells. And again, we can see here that the complex produce minimum mobility. So inhibit the mobility of the cancer cells when we compare to, for example, the positive control or to the negative control. We can see here. And here also very interesting, when we use only the FGF, the maximum mobility, you can see it's FGF. It can facilitate, can activate cell signals and resulted in activation of cellular growth, repair, and other cellular properties. So here we can see the complex also inhibit the mobility of the cancer cells. And again, we assume or predicted that this is one of the causes of the improvement of the activity or overall activity of the targeting complex. Another study was conducted is to study the inhibitory effect of the complex on the colony formation of the cancer cells. And again, here, this is also another study to predict how this complex could inhibit the growth of the cancer cells or the possibility of also causing a cell death. And again, here we can see the complex resulted in the highest inhibition to the formation of colonies compared to the pure drug, beta-cyclodextrin, lactose also used here as a control, and also FGF again here resulted in the maximum colony formation, even when compared to the negative control. Again, this is also another study that can support that this complex can inhibit or can produce better cytotoxic activity when compared to the pure drug. The last part was to use the capsule as also a targeting technique to the colon, because here not just targeting the cancer cells and also the site of the action. Here we choose these types of coating, which is interior coating, because it's known to only dissolve in the colon due to the pH of the colon. So that coating will only be dissolved in the colon pH. Then the capsule will be ruptured and release the content. So this was tested using animal model. As we can see here, the left side, we use uncoated capsule first to test so that capsule will reach the colon or not. And as we can see here, it starts rupturing from the stomach and releasing its content. And it was not ideal. So then we tested with the coating. And as we can see here, minimum or no release at all of the content of the capsule at the stomach, and it's only released after the stomach, because at the pH above 6, the coating will start to dissolve, and the capsule will also dissolve or rupture, then the content will be released. So here is the idea here is that this dosage form would be utilized to carry or the complex to that site of action, or the site of the targeted site, to release the drug and to also help with the targeting process, the overall targeting process. And by this also, we ensure that this complex can be taken orally. This is very important, because this is also could relieve many effects or unwanted effects related to injectable therapeutics. So here we improve the solubility, and also able to deliver this formulation orally. This is also another advantage of utilizing this technique, targeting technique, and the complexation of everolimus with beta cyclodextrin. So in conclusion, we can say that the complex show the better activity than the pure drug, and also effectively and targeted cancer cells more than the normal cells. And this is also tested by treating the normal cells with the complex and compare its activity with the cancer cells and normal cells, and we found that the selectivity index is more than 10, and this is good because it showed that the complex having more selectivity to the cancer cells, and also shown that it could inhibit the motility, migration, colony formation of the cancer cells, and all these contribute to its higher activity and cytotoxicity towards cancer cells. And also this work was awarded a silver medal when it presented in Malaysian Technology Expo in 2020, and we were also able to publish several articles in ACS Omega Scientific Reports and Open Nano. So here we can say that the targeting technique, this targeting technique was successful to deliver the or to target the cancer cells instead of normal cells, and improve the overall activity, and possibly reduce its severe effect or adverse effect known to be associated with cancer therapeutics. By this, I come to the end, and thank you very much for listening. And please, if you have any question, you can send to me, and I will respond to it. Thank you. Thank you so much.